Hi, everybody, and welcome to this session of Sigma Xi's Hangouts with our distinguished lecturers. I'm Fenella Saunders. I'm the managing editor of American Scientist magazine. Thank you very much for joining us. This particular session is sponsored by the RTP chapter of Sigma Xi, so thank you to those members and welcome. And I just want to mention before we get started, the next one in this series will be on February 4th. We'll have a hangout with Todd Suravel about some anthropology topics in Mongolia, so that should be a lot of fun. Please plan on coming back for that. We'll also have a Google Hangout in January related to an article coming out in American Scientist on fire in space, so please look out for that as well. The next round of Sigma Xi Distinguished Lecturers for the 2016-2017 season will be announced in the next issue of American Scientist, which is coming out imminently, so please also keep an eye out for that. And please have your chapters join in and let us know if there are any of these hangouts that you'd like to be involved with. So today, we're very lucky to have a hangout with Dr. Bryant Nelson of the National Institute of Standards and Technology. We're going to be talking about nanoparticles and how they interact with DNA. If you have questions during the session, you can submit them either on the event page or you can submit them in the hangout here. There is a Q&A on the right side of the screen. If you don't see that, there's some icons at the top of the screen that'll let you turn that on. If you are a Sigma Xi member, you can go to the Sigma Xi Communities page after the session and ask questions there. We'll follow up with Dr. Nelson with those questions. So thank you very much, Dr. Nelson, for joining us.
Thank you for having me.
Okay, this is very exciting for me. I'm definitely a fan of the nano world myself. So, but I know there are people who are going to be joining us who don't necessarily understand what nanomaterials are. So I wondered if we could start out by talking about sort of what the definition of a nanoparticle is. What sort of size range are we talking about?
So one of the definitions for nanomaterial, believe me, this hasn't been settled by a long shot, but one of the definitions for a nanomaterial, engineered nanomaterial, is a material that has a size range between 1 and 100 nanometers. So there's a large debate going on around the world about whether or not that size range should be raised up to about 1,000 nanometers, because until you reach 1,000, you're not at 1 micron. So, but that can be discussed in many different levels. But nanomaterials in general are unique materials that can have really exciting properties in terms of their catalytic abilities, abilities to interact with biological molecules. They can induce high levels of different types of increases in strength to materials. So it goes on and on. Engineered nanoparticles are basically always around us. They've been here since time memorial. So, but we're calling them right now engineered nanomaterials when they actually have been around as natural nanomaterials for a very long time.
Right, okay. So is there a way that we can give people a sense of the scale that we're talking about here? I mean, this is very small, but is there a way for that people can think about the kind of scale where I know a lot of people compare this to sort of the width of a hair. Are there other things that we can give people a sense of here?
Sure. You can think about proteins, for example, or DNA, which we're going to discuss today. So DNA, our DNA in our bodies has a diameter about 2 nanometers. So that's on the scale of nanomaterials. You can't see them with the naked eye. So that's basically the area that you should concentrate on when you're thinking about nanomaterials.
Right, okay. And you mentioned that these materials have been around for a long time, but now there's sort of this distinction between natural and engineered. I wondered if you could talk a little bit about a natural nanomaterials. Are there some examples there that we can give people of what could sort of be a natural nanomaterial?
Sure. Really good examples are these carbon nanotubes. So carbon nanotubes are prevalent in the environment, and the initial forms of these materials come from like volcanic eruptions, or even from exhaust from automobiles. But we can design these carbon nanotubes using chemical procedures or chemical vapor deposition to produce engineered carbon nanotubes.
Right, okay. And are natural nanoparticles or nanomaterials sort of inherently damaging, or are they benign, or is there sort of a mix of the two?
Natural nanomaterials are what they are. They exist in the environment. You can't say whether they're damaging or are not damaging. They just exist.
Right,
It
okay.
depends on
So
the circumstances.
right. It's sort of one of those case by case basis.
Very
Something.
much so. Yes.
Yeah. Okay. One thing that I wondered if you could touch on for a moment before we get to your specific research is, within the last couple of decades, there's really been this sort of explosion in production of nanomaterials, and I wondered if you could kind of touch on why that might be. Is that just increased technology, or have people discovered new uses of them? Is it more of a fad?
Well, it's definitely not a fad. There's a lot of commercial interests in the production of nanomaterials. I think that's one of the main drivers. Another driver would be the innovation in technology. You can produce new and better types of and stronger materials out of certain nanomaterials. And there are some nanomaterials that may have some, I would say, medical applications. Just to give you an example, silver, the precious metal, can be engineered into the form of a nanomaterial, silver nanoparticles. And there's a lot of studies out there that show that silver nanoparticles may have some type of antibacterial properties. So the commercial products that contain silver nanoparticles include some socks. There's even different types of, I guess, washing machines that have silver nanoparticles built into their components. And the reasons for this is because these silver particles have been shown to impart some antibacterial properties. And so, for example, for socks, you want its antibacterial properties to reduce the smell from bacteria. And for washing machines, you want to increase the level of cleanliness of your clothing during the washing cycle.
Right. And actually, one of the things that I think people are becoming more aware of is that as these products are getting to be more in the consumer mainstream, you're getting a larger release of these nanoparticles into the environment. Is that one of the basis for your research in terms of nanoparticles having more ability to interact on a cellular level? And is that just an exposure increase?
Exactly. We want to know what type and how much of the nanomaterials are potentially entering the environment. So, but in order to do that, we have to have the methodology and measurements and protocols in place to be able to actually measure the small quantities that are released and to identify the type of nanomaterials that are present. It becomes really complex in that there's just not going to be just one potential type of nanoparticle or nanomaterial released into the environment. There's going to be multiple different forms, and so we need new methods and technologies to be able to discriminate between all these different types of materials.
Right, okay. And one of the things that I was curious about, you had mentioned carbon nanotubes before. Those are often very long molecules, comparatively speaking. And I know that there have been studies comparing them and their effects on, say, lung tissue, comparing that to potentially the way that asbestos can damage lung tissue. But I'm thinking that what you're looking at is even on the smaller scale, because you're talking about them interacting with DNA at a lower level than, a lower size level than even on a sort of a cellular tissue level.
Yes, we're looking at the molecular level interactions.
Right, okay.
Yes, we're trying to understand how these molecules and, for example, how carbon nanotubes may change the properties of DNA.
Mm-hmm.
So we're developing, our focus is to develop new, better measurements and methods that can allow us to explore, investigate those interactions with DNA.
Right, okay. We've just had a question from the audience come up, and sort of before we move on to the DNA, we'll just
Sure.
clear this one up. There's a viewer who asks, silver nanoparticles have also been shown to be destructive to the environment, to waterways and fish. And she's just wondering about impacts on the environment and even on the microbiome from things such as silver nanoparticles. Could you just speak about that for a couple of seconds?
Actually, so what we do at NIST is we actually develop methods for detecting, for example, those silver nanoparticles in different environmental matrices. So we can develop methods that are capable of detecting single silver nanoparticles in different types of matrices, whether it's like a water matrix out in the environment, or in soil or sediments, or even in the air. So, but the methods that we develop are only used by other scientists to actually look at, I would say, the effects, the biological effects, or the effects on the environment. We only focus on developing really robust and reproducible methods. We don't actually try to understand and discriminate the different types of responses.
Right, that makes sense. I mean, basically, you have to be able to quantify what's there before you can determine what kind of effect it has.
Yes, and we leave those types of studies to other institutes.
Right, okay. So if we'll kind of turn here to talk about the work that you specifically do with the DNA interaction, but just to give people a little bit of background on that, I wondered if we could talk about DNA's ability to repair sort of in general first. Can you talk about why it needs to repair, how resilient it is when it repairs, things like that, just sort of generally why DNA would need to repair in general? Why would it have that ability?
Oh, certainly. Well, our bodies, of course, contain DNA in all of the cells. And so even when we're just walking around, our DNA is being damaged by both endogenous and exogenous sources. For example, UV light can induce some damage to our DNA, and therefore the body has to have a mechanism for repairing that damage. Fortunately, humans and other living organisms have many redundant DNA repair systems in the bodies. So one of these systems is called base excision repair, and this type of repair is used to repair damage from like ionizing radiation. So if our cells encounter any type of ionizing radiation, the redundant repair system kicks in and repairs that type of damage. And this damage is usually oxidative damage. So your DNA may be oxidatively modified. And if your repair system is working correctly, then it will, of course, kick in and start repairing those damaged sources of DNA.
Okay, so let's talk about that oxidative damage just for another minute or two, because that actually comes up in the research that you do as a mechanism. So when you're talking about oxidative damage, what's actually causing that damage? Are we talking about free radicals here?
Yes, exactly. So both inside and outside our body, our DNA and cells are constantly bombarded by free radicals. And so free radicals such as hydroxyl radical, superoxide, and even non-radical species such as hydrogen peroxide. They can, if they're close enough to the DNA, they can actually interact with the DNA in the cell nucleus and cause these changes, oxidation to the DNA bases.
So let's just help people who might not remember too much about oxidation from when they last studied it. Can we talk about what actually changes within the DNA when oxidation occurs?
Oh, sure. So in our case with ionizing radiation or any other type of chemical inducer, what happens is, for example, and you have a guanine base, one of the 4 bases in DNA, it may encounter, for example, a chemical compound that produces hydroxyl radical.
Mm-hmm.
If that happens, then the guanine is oxidized at a certain point on the molecule where you have an addition of a hydroxyl radical. That's oxidation.
So it actually attaches to the molecule.
It actually attaches to the molecule.
Mm-hmm.
So you can have that type of oxidation across a double bond, or you can actually have oxidation followed by changes in the structure, where the structure of a given base may either collapse or expand. So there's different types of oxidation that occur. You can have addition of say a hydroxyl radical to the given base, or you can have you can cause protein DNA crosslinks, for example. You can cause the DNA to lose a base. Actually, this is called induction of an apurinic site, where you actually, the DNA or the base chain has different sites that are missing bases.
I see. So it's not just, excuse me, it's not just losing bases, but changing the properties of the base is a potential outcome as well?
Exactly, yes. You change the properties of the base. And so when you induce these oxidatively modified bases, or lesions, however you want to call them, the sequence of your DNA is modified. And so if those bases, for example, if they become set, if your cells replicate and those modified bases become set, then you have the increased chance of mutagenesis. Okay, and then further from that, of course, that could lead to cancer.
Right, okay. So one of the things that I thought we should maybe just mention here is obviously there are disease implications here, as you just mentioned, but this kind of mutation over evolutionary time can potentially be advantageous as well in the long run.
That's true, yes. So there's it's the face of Janus. You have the good and the bad.
Yeah, that makes sense. So let's get back to what you're doing more specifically here now that we have that groundwork, and let's talk about how the nanoparticles might actually cause this kind of DNA damage. Do they actually get into the DNA themselves, or do they have sort of a chain of events that leads up to that damage?
There's several different mechanisms that have been identified for how nanoparticles could potentially cause DNA damage, or cause or enhance DNA repair. Actually, one of the predominant mechanisms is through nanoparticles producing free radicals, such as hydroxyl radical. So some nanoparticles, if they're based on, say, metals like iron oxide or copper oxide nanoparticles, they have that transition metal in there, then they can produce free radicals based on the Fenton reaction. So that's simply, you have hydrogen peroxide in every cell in the body. If nanoparticles are able to enter that a given cell and actually enter the cell, interact with the hydrogen peroxide, if the hydrogen peroxide then converts to, well, the hydrogen peroxide actually can convert to hydroxyl radical, enter the nucleus, and then you have potentially DNA damage.
Okay, so how do nanoparticles actually get inside cells? Are they just transported in through sort of standard portals, or are they fooling the cell into thinking there's something else?
Some nanoparticles actually can enter cells through endocytosis. The mechanism is not the same for every type of cell. There's different mechanisms based on the cell type. But one of the most common ones is endocytosis. So that's one way.
So that's where they just kind of get engulfed within the cell itself, like through sort of a vesicle in the wall?
Yes, and then at that point, they usually end up in the lysosomes of the cell, cytoplasm, and then broken down. And then if they break down into certain types of ions, for example, metal nanoparticles, again, those metal nanoparticle ions can interact with hydrogen peroxide
Right.
and
Okay.
form hydroxyl radicals.
And you're saying this is just one of the mechanisms that's possible. I think
Yes,
that
yes.
there's another one that involves inflammation. Is that
That's
correct?
that's another one, but there's even another one where
Okay.
the nanoparticles that actually, if they're small enough and can enter the nuclear pore of the nucleus and get transported via nuclear pore proteins into the nucleus, they can actually interact directly with DNA.
I see.
But then,
So
yes, there's another mechanism where nanoparticles enter the cell in general. If enough of them enter, they can cause an increase in oxidative stress in the cell. At that point, the cell recruits different types of neutrophils and macrophages to the spot in the cell that has the nanoparticles, and you get enhanced production of free radicals at that spot, which can travel throughout the cell.
I see. So the inflammation response actually produces the free
Yes,
radicals?
exactly.
Okay.
And
And if we
that's actually a natural response to an exogenous particle that's entering the cell.
mhm.
They're
So
trying to you know, a type of repair mechanism.
so in a way they're sort of hijacking this natural repair mechanism and causing damage or
Well,
inadvertently?
they're actually, what's going on is they're exceeding the natural antioxidant defense system of the cell.
I see.
There's so much ROS being produced that the natural protein and small molecule antioxidants in the cell can't handle the increased level. And so you get this oxidative burst, and that may or may not overwhelm the cells.
I see. Okay. Now, just to go back 1 second to the other mechanism you're talking about, where you said that they could actually interact directly if they're small enough, if they could interact directly with the DNA. So what do
Sure.
they do in that case? Do they actually sort of cause these cuts in the DNA itself, or?
Or they can if they are still oxidative, if the properties of the nanoparticles are such that they can produce free radicals. But I think the main mechanism may be that the particles actually bind to the DNA inside the nucleus, and of course that causes all kinds of havoc.
Right. Okay.
It potentially cause, yes.
Okay. Um, I don't want to interrupt, but I think maybe your office light has gone off. I've had that happen to me before during interviews. I didn't know if you wanted to try to get it to go back on, and then we could see you a little better.
Okay,
So
is that better?
yeah, that's great. Yeah. So I've had that happen too. These automatic lights want you to move around more maybe than you are when you're on one of these things. But
Yes.
thank you.
Thank you.
So one of the things that I wanted to talk about is, you are at NIST, and I wondered if is your main concern figuring out how to quantify this damage that happens to the DNA?
My main concern is to actually develop methods that can be used to quantify these types of damages to DNA. So we're trying to develop robust, very sensitive methods that can be used by others to, to actually look at the potential bioresponses. But our focus is to, develop robust methods that
Right.
are highly quantitative and that also can identify the particles that are present. So we have two main strategies: identification of the particles and quantitation of the particles.
Right. Okay.
And the focus these days is really to be able to identify single particles, individual particles in different types of matrices. So we're always pushing the limits of our capabilities and what we're trying to reach to more sensitive analytical tools that are robust and reliable.
It seems kind of incredible to me that these mechanisms could be sensitive enough to identify single particles. Is that really the level we're at, or is that just the goal?
No, we're for certain types of measurements, we're at single particle levels, yes.
Wow, that's really amazing. So when you're actually developing these methods, is it already known sort of the locations on the DNA where these sorts of damages occur, or is that sort of like what you had to develop first?
Well, the damage can be at any location on the DNA. So any of the 4 bases or even the phosphate backbone. And a lot of the methods were, well, not a lot of them, but the background of some of the methods were already in place to try to transfer those methods for nanoparticle measurements. So we had a lot of methods already in place based on mass spectrometry for chemical induced damage to DNA. And so we made modifications to those methods in terms of how to reliably extract DNA from plants and organisms, and then measure the amount of damage induced by certain types of nanoparticles.
Right. Yeah, I want to kind of break both of those things down a little bit here. So you're talking about extracting DNA. So I guess you're using several different models to try to do these tests. Is that correct?
That's correct. Yes. So we're using the most simplest model up to most complex. So we increase the biological complexity of our studies over time to try to really understand how our methods are performing. So we start with the simple DNA model, where this DNA by itself, no cells, no tissues, no organs from organisms, and we incubate that DNA, calf-thymus DNA, with nanoparticles. And we measure how, we try to measure the production of these DNA modifications. So to get a baseline for how nanoparticles may or may not interact with DNA. From there, we move on to more complex systems, for example, involving cells. So now we have to develop procedures to extract the DNA from cells without causing modifications to the DNA, and then measure the types of bases that are damaged. And then finally, the ultimate goal is to develop methods where we can extract DNA from higher organisms, like plants and like C. elegans, for example, that have been exposed to these nanoparticles. Extract that DNA and look at how what types of modifications have occurred in that scenario. So 3 different types of exposure scenarios is what we're talking about.
So are you currently using all 3 of those methods, or is that something that you're kind of working up to?
We're currently using all 3, yes.
Okay.
You get the most information from many different sources. So we put all that data together and look at how our methods are performing.
Right, okay. So when you're actually looking at those different methods, are you actually using different levels of incubation with each of those cases? So you're sort of measuring different exposures, or do you have kind of a standard exposure that you're using across those cases?
Oh yeah, we have a standard exposure scenario. So we have incubations, exposures to nanoparticles. First of all, before we even get to the exposures, what we really need to do is work with well-characterized nanoparticles. So at NIST, we have these things called standard reference materials. Within that program, we have standard reference nanoparticles. So these are materials that are completely well-characterized in terms of their physical and chemical properties. And so with the nanoparticles that we work with, we use these reference materials as our baseline. And then we look at how other commercially important nanoparticles compare to those reference materials. So we may work with collaborators both inside and outside of NIST who want to look at the effects of their particular nanoparticle on a certain model. And so what we do have defined protocols in place where we treat all the nanoparticles exactly the same throughout the exposure scenario based upon our reference nanoparticle.
Right, okay. Okay, so Again, just to go back to what you were talking about before, to try to break this out a little more, you were talking about some of the mechanisms that you're actually using to develop these quantifications. And you mentioned, first of all, mass spectrometry. I wondered if we could kind of just do a little intro on that for people who aren't familiar with how that works.
Oh, certainly. So we're actually employing two different types of mass spectrometry, most of them based on isotope dilution. This is where you take a known chemical that has been stably isotopically labeled, with example with C13 labels, and you incubate that with the analyte or whatever compound you're trying to measure. You use the ratio of the signal responses to actually do your quantification of the sample. But anyway, we're using isotope dilution mass spec in terms of gas chromatography and also in liquid chromatography as a separation up front. And we are actually measuring all the DNA bases that currently measuring all the DNA bases based upon extraction from a given matrix, adding in an isotopically modified base, isotope labeled modified base that we know, and then measuring the output using electrospray ionization mass spectrometry, which is a type of mass spec that where you actually generate ions of your analyte and measure it based upon that ratio of to the unlabeled analyte.
So for people who just aren't as familiar with the method, I mean, we're talking about measuring a spectrum, but what is the spectrum actually measuring there? You're measuring different levels of a particular chemical?
You're actually measuring the different bases of DNA, the adenine, thymine, guanine, cytosine levels. So you have your the bases that are modified, oxidatively modified, and you want to know how many of each base is present based on a given amount of DNA. So, like I said previously, you add in a known amount of a labeled version of that modified base, and then you can take the ratio of those of the bases that are modified in your DNA sample, compare it to the ratio of to the amount of DNA that you add in, and then you can do your quantification that way. So we're actually measuring the different bases that are in the DNA molecule that are modified.
Okay.
And so we have to actually, to get those bases, we have to remove those bases from the DNA chain. And the way we do that is we take a sample and we incubate that sample with a base excision repair protein that is only specific for certain types of bases. And it chops out the bases, removes those bases from the chain. So we have those bases in solution, and therefore we can measure how much was present on the molecule, the entire DNA molecule.
Right, that makes sense. And so is it actually measuring sort of the chemical property of the base? What is it actually using?
So the mass spec actually measures the mass to charge ratio of each species that is in the sample. So in the end, we come up with a mass of how much of each base that is in that sample.
Right, and so the masses are very specific for each one of these bases.
Yes.
Right, so it's almost like a fingerprint for the base in that sense.
It's more than a fingerprint. It's actually an absolute identification of the base. Because we always use standards to identify which base is present in any given sample.
And you also mentioned gas chromatography in there. I wondered if we could sort of give people a little bit of an idea of how that works.
Okay, so the gas chromatography or the liquid chromatography, we use either one of those techniques as a separation tool. And so gas separates molecules that are in the gas phase, and liquids separate molecules that are in the liquid phase. And so it's based on injecting your sample into either the gas chromatograph, where then it's vaporized and then ionized, and the ions are separated, are actually analyzed by the mass spectrometer.
Okay, so that's basically the way that you can kind of separate out the different things that you're getting in this sample.
That's correct. Yeah, we can either use, like I said, liquid chromatography or gas chromatography to separate the species in the DNA sample.
Right. Okay. And you mentioned that these were the baseline mechanisms that were in place. How have you actually had to modify these methods to do the kind of research that you want to do?
Well, first of all, we had to modify the extraction procedures to make sure that the samples that we are working with, that the nanoparticles are actually isolated from the system, because we don't want to introduce any nanoparticles into the mass spectrometer. So we had to come up with specific extraction procedures to isolate the DNA from the nanoparticles. And then we had to fine-tune I would say, the detection parameters for the DNA bases so that they were more sensitive for the low levels of bases that we're trying to detect.
Mm-hmm. Is there, are you actually worried about the sample size at all? I mean, I'm wondering if this is something like, where you have to use something like PCR to increase your sample size, or if a small sample size doesn't matter to this kind of detection mechanism.
Well, actually, in this detection mechanism, we're kind of worried, not worried, but concerned with the sample size, because we require a lot of sample. For all the mass spectrometry based techniques, we need a large amount of DNA, so on the order of 50 micrograms. So most techniques, of course, need nanograms, but the information that we get from this technique based on mass spec is tremendously powerful. So it's a trade-off. We need more sample, but we get more information.
Right. Okay.
So we're concerned about not having enough sample.
Right. Okay. So when you're saying 50 micrograms there, can you give us a sense of how much that is? How many cells of DNA is that equivalent to, or something like that?
Oh, yeah, sure. So for example, if we were talking about HeLa cells, that would be roughly 5 million HeLa cells.
Wow.
Will give us 50 micrograms of DNA. If we're talking about an organism such as C. elegans, we would need about 250,000 C. elegans worms for 50 micrograms of DNA.
Wow.
But C. elegans grow like wildfire. We get billions of them. We're culturing those in the laboratory. So we really don't have a shortage of DNA. It's just nice to have, not to have to worry about that.
Right. So it's more a question of making sure you have a sufficient amount of culture
Yes,
exposure going on so that you have the right amount of sample size.
that's correct. Yes.
Okay.
And so one of the things that I like to talk about is, there's a lot of studies that do these types of exposures. A lot of researchers, but reproducibility is really key. And so I think that we try to focus our laboratory so that we do every experiment either 2 or 3 times. I mean, the entire experiment, the exposure, because you might see an effect when you do something once, but then it's not reproduced the second time you do it. So you have to do them several times to be sure that what you see or what you measure is real. And that's a big problem in science right now. I'm sure you've heard. It started out in I think there was rumors in pharmaceutical or clinical oncology industry where a lot of studies have been produced and put out into the public, but then when industries had tried to reproduce those studies to actually manufacture drugs, the studies have failed. And so at NIST, I know we're trying to make sure to get the message out that studies in general, where you see a response, whether it's positive or negative, you really need to do that study several times to verify the validity of that response. And so, yeah, that's one of the points that I wanted to make during this conversation, is that reproducibility is key.
Right. Yeah. No, that totally makes sense. And there have been a lot of high visibility stories about
Yes,
retractions based on
yes,
lack of
exactly.
reproducibility in the last few years. So I understand why, especially someplace like NIST, which is all about standards, how you would want to have reproducibility sort of locked in everything that you do.
Yes, and that's especially so with our exposures, we don't just do them once. We do them many times to verify, uh, and just to verify the experimental conditions too are reproducible, so that the methods that we put out there, others can rely on them to give them ground truth, or answers that they can trace back to a given method that has been shown to be reliable.
Mm-hmm. I just had a quick question, though, going back to your exposures that you're talking about. If you're sort of putting DNA directly into a concentrated solution of nanoparticles, is that kind of a worst case scenario, or are there actually concerns here about exposure levels that people might experience naturally within a standard environment?
No, I wouldn't say it's a worst case scenario. I say it's a case where we are trying to understand the mechanism. So if we have a lot of DNA, if we have a known amount of DNA that's uncontaminated with other species, where we can actually just hone in on each base specifically, we can get a better understanding of how nanoparticles are interacting with the DNA. So no, it's not the worst case scenario. It's just a convenient exposure media.
Right, so it's more establishing the mechanism rather than the level of exposure necessary to produce that response?
I think it's 50-50, because we're looking at both aspects of that. We're trying to look for whether or not our exposures can potentially introduce artifacts into our measurements. And so if you just have the DNA by itself, you can really identify, is this an artifact, this lesion that forms because we're using, like, for example, really high levels of nanoparticles, or is it a real effect based upon the nanoparticle producing a free radical or binding to the major or minor group of the DNA? Things like that. We can
Right.
discriminate between, whether it's a real effect from the exposure or from the nanoparticle.
Right. Okay, that makes sense. I'm assuming that you've obviously found positive correlation between nanoparticle exposure and DNA damage. I wonder if you could kind of summarize some of your latest results in that area.
Well, some of the results that we've seen, for example, actually it's an artifactual um, um, study, that not an artifactual study. We were looking at the effects of potential artifacts from exposures. And one study that we looked at was you might have heard about titanium dioxide nanoparticles. They're used in paints, they're used in cosmetics and various other consumer products. Some studies have shown that the titanium dioxide, when exposed to light, can produce free radicals. And other studies have shown that it doesn't produce free radicals. And so we were interested in, developing like an exposure scenario where we could investigate whether or not it was true or not, from the effects of titanium dioxide in light. So most of those studies were based on the use of the COMET assay, which is a single cell gel electrophoresis assay that separates where you can measure DNA that has strand breaks in it based upon electrophoresis of the DNA in a gel. Well, anyway, we designed a study where we expose DNA, just plain DNA, with titanium dioxide nanoparticles under different conditions of light. No light, regular lab light, for example, and UV light. And we measured the amount of DNA lesions that were produced. And so we did this all under very controlled conditions, where we knew how much light was in the exposure, we knew the temperature, and we knew the exposure concentrations at infinitum, okay? And so what we found using our mass spec procedures during this exposure was that DNA and titanium dioxide and DNA in the dark does not induce DNA damage in terms of oxidative damage. In the light, it depends on how much titanium dioxide is present. Visible light, you can see some DNA damage. And with UV light, you can see a higher level of DNA damage. So the study showed basically that you have to use controlled conditions, measurement conditions, where you know exactly how much light is being exposed to the DNA and the nanoparticle. And then you can make an assessment of whether or not DNA damage is occurring. So this is basically, we have devised a protocol for how you can actually determine whether or not DNA damage is occurring using in the presence of titanium dioxide nanoparticles.
Right. Okay. That's really fascinating. But you know, we're getting a little short on time, so I want to hit on the other face of Janus that you talked about earlier. And
Okay.
you know, we were mentioning that nanoparticles may actually help with DNA repair or reducing DNA damage under certain circumstances, or fighting cancer and things of that nature. So maybe we could talk about that side of things a little bit. I don't know if you want to start with this, but I know that one of the things that you've looked at is that single-walled carbon nanotubes can actually kind of scavenge up free radicals under certain circumstances as well, and kind of prevent damage in that sense. I didn't know if you wanted to touch on that.
Oh yeah, so actually there have been other studies that have shown that single-walled carbon nanotubes to distinguish from multi-walled carbon nanotubes. So actually both, now that I think about it, have shown that they can actually scavenge free radicals in various different types of cell environments. So yeah, there's studies going back to at least 2003 that have shown that carbon nanotubes can scavenge free radicals in cell studies. But we wanted to try to develop a method where we could determine whether or not carbon nanotubes could, if they can scavenge free radicals, can they also prevent DNA damage or reduce DNA damage? Since free radicals is a known cause of DNA damage, oxidative DNA damage. So we designed an exposure study where we took a mixture of carbon nanotubes, single-walled carbon nanotubes, and we exposed them to sonication conditions and also DNA. And the way we did that was we took the carbon nanotubes and wrapped them with single-stranded DNA and exposed them to a defined amount of ultrasound. Now, ultrasound is known to produce free radicals similar to ionizing radiation. You get the same sort of suite of free radical production. Hydroxyl radicals, superoxide, hydrated electrons, and hydrogen atoms produced. And so we knew that going in that a high level, if you expose these single-walled carbon nanotube, the carbon nanotubes wrapped with DNA to the ultrasound, you get a lot of free radical attack. But we wanted to see whether or not the carbon nanotubes had an effect on scavenging those free radicals. What we found out was yes, indeed. So if we took, utilizing two different types of DNA oligos, we've shown that you can reduce the level of DNA damage based upon the carbon nanotubes' ability to either scavenge hydroxyl radicals or scavenge hydrated electrons. And so, yeah, in this case, we've shown using a cellular model and a defined exposure system that carbon nanotubes can actually prevent DNA damage. Now, the next step, though, is for other researchers to use this methodology and try to determine whether or not this can actually occur in like a cell system or in the organism model.
Right, so to actually turn this into some kind of treatable therapy or something like that is a very big step.
Yeah, it's a huge step. There's a lot that needs to happen before that can occur. But at least we've developed a method where you can actually measure this occurrence, this type of radical scavenging from the carbon nanotubes.
Right, okay. And I think that you had also mentioned there are certain kinds of possibly metallic nanoparticles that can be used in ways to increase things like drug targeting or cancer therapy. I wondered if you could talk about those things as well.
Yeah, that's work that's ongoing. There are some that we have some information about really small nanoparticles. Remember at the beginning I defined nanoparticles as being the size range that's lying between 1 and 100 nanometers as one definition of a nanomaterial. We're working with these really small, like 1.4 nanometer gold nanoparticles. And we're trying to actually determine, develop a procedure to figure out whether or not these particles can inhibit DNA repair proteins. So what we're trying to do in this particular circumstance is take known DNA repair proteins that are well characterized and expose them with the nanoparticles in solution and look at the effects on the DNA repair excision activity of those nanoparticles. Thus far, we've seen that those particles at that size range of 1.4, these are gold nanoparticles, by the way, that they are showing some evidence that they can inhibit DNA repair.
So in that case, you're talking about inhibiting DNA repair. Like if you could target that to, say, a cancerous cell, you could use that to disrupt the cell.
If you could target that to a cancer cell, then you may have some groundwork to further develop that as a potential therapy for different, as a modality for anti-cancer agents.
Right, and I think you may have mentioned, correct me if I'm wrong, but the fact that these are metallic nanoparticles means that they could be potentially directed with magnetism or some other methods to kind of target them to certain cells.
Oh, yeah, you could actually coat these particular nanoparticles with different types of receptors. If you could figure out the correct receptors for different types of cancer. You could coat them with receptors, and you could coat them with different targeting ligands. And yeah, there's many different ways to expand upon the use of these nanoparticles. But that's a tremendous amount of work that needs to go into that.
So are you actively trying to develop specific nanomaterials that can interrupt DNA repair at this point, or are you kind of working with existing ones?
I'm looking at existing ones right now. I'm not developing. I'm not in the. That's not my area of expertise. Developing new nanomaterials. We have other groups at NIST that actually look into that and work quite heavily in that area. But I'm, like I said, I'm trying to focus mainly on developing new methodology, new technologies that these other groups can use to actually, verify the identity and the level, the types of their nanoparticles that they're creating, the location of these nanoparticles in whatever model that they're trying to use, and also to be able to quantify the level of those nanoparticles.
Right, okay. We've got about 5 minutes left, so I just want to take a question here from the audience, which is a little bit more on a general level, but maybe you have some opinion on it. The viewer's asking, it seems like policy is far behind research and development on nanoparticles. Most people don't even understand what they are, and they could get a bad rap if the public starts seeing them as carcinogenic. Are there any policy implications for your research in that sense?
Development of methodology. I think that as long as NIST and my group in particular continues to develop robust methodology that is highly sensitive for measurements, and that is reproducible for others to use to actually determine whether or not these nanoparticles are hazardous or good for the environment or for human health, then we're doing our mission. We're on track. NIST doesn't get involved in policy making or policy decisions, just methodology.
Right, okay. So you just mentioned before that you're working with other departments at NIST, and I'm assuming that some of those collaborators are in different fields than you're in. I was wondering if you kind of talk generally about, do you feel that cross-disciplinarity is helpful in research? Is that something that you strive for in what you do?
We couldn't do it without being cross-disciplinary. I work with physicists, environmental engineers, biologists, biochemists, and material scientists. We talk daily about the types of experiments and results that we generate. And so it is absolutely crucial to be able to bring together a diverse group of scientists and engineers in the area of nanotechnology, because it is changing faster than we were able to create methods to measure these new materials and to determine how they're going to behave in the environment.
Right, okay. And you mentioned a diversity of teams there. I wondered if you'd had some experience with this. It's sort of generally accepted that diverse teams in science produce better results. I wondered if you could talk sort of about your experience and if you feel that diversity amongst the scientific team can increase your creativity and your actual achievements.
Absolutely. That goes without having really, you can't be innovative in this environment where funding is so tight, without working in, I don't want to say too large of a group, but at least in a group environment where ideas are able to flow freely, and you may not, be able to understand all the different ideas, but if they're on the table, you can have a good, honest discussion about the differences and move the projects forward, move science forward. So what I mean is that you can't be an expert in every area. So you do need have to have an expert on the team though, in that particular area or any particular area. So yes, you can't really go forward rapidly without having a diverse team.
Hmm. And do you think having scientists from different backgrounds, from different nationalities, from different ethnicities is helpful in developing different ideas and concepts?
Absolutely. And at NIST, we try to have that type of I guess, mentality, and also um, we always want to have a diverse team as possible. Because in scientists, you can't, I mean, in science in general, you can't discriminate. The science itself will discriminate at the end of the day.
Right. That makes a certain amount of sense, of course. So as somebody who works at NIST, which is not sort of a traditional university setting, I'm wondering if you feel that scientists, especially people who are newly graduating, are often looking at alternate paths than academia. Do you think that right now there are more opportunities for recent graduates or other scientists to kind of branch out from the traditional academic routes? And can you talk about some of the best ways to help graduates find meaningful alternate paths?
Okay, the best way, well, I'll start at the end. The best way to find meaningful alternate paths is to network. So there are a number of different, I would say, meetings around the U.S. that are geared toward bringing together a diverse group of scientists for just networking. There's like the organization for the SACNAS and Novache, and going to those types of big meetings, even also the ACS, you get to meet your peers who are already working in maybe areas that you want to go into. So then you can have face-to-face time with those peers. They get to know who you are on a first-name basis, and then you may have an easier time entering, if it's academia you choose, academia or industry or even government. But I think face time is critical. NIST is, as an institution, is a mix between academia and industry. Even though we're a government institute, it's an open environment, and we collaborate quite widely, both domestically and internationally. So we get to know a lot of different folks. And I think this is one of the best places to, for me to have established a good career. And I wouldn't change where I ended up.
Right. We're running just a couple minutes over, but I did want to throw in just one final question for the Sigma Xi members out there, because I know you're a past officer of your own Sigma Xi chapter there at NIST, and I am also an officer in the RTP chapter here. And
Okay.
so just as somebody who's been involved at that chapter level, I know one of the things that often is a challenge for chapter leadership is maintaining engagement with the members in their chapters. I wondered if you had any general advice from your experience as a chapter officer for other chapter officers on some of the best mechanisms for engaging with their membership.
Happy hour. No. Yes, NIST has a very strong chapter, and I think that our I guess our most used mode of staying engaged is to have monthly meetings, where the officers and also the members meet to discuss what's on the agenda, what's coming up short time in advance and also on the long time horizon. And I think, FaceTime is key. We're all inundated with too many emails, and it's so easy to click to the trash or to clutter, that you have to rely now on FaceTime. And I would recommend that we eliminate, as many of the email purges, emails going out, and just set up a meeting with all the members.
Yeah, right. Sort of a return to the roots of the whole point of a society.
Return.
Right. Right.
Yes.
Makes sense. Okay, well, I don't want to keep you any longer because I've already run a few minutes over, but I do want to thank you so much again for sharing your time with us, and thank you to everybody for watching.
I really appreciate this, and thanks for the invitation, and I hope it was useful.
Thank you so much, Dr. Nelson.
Thank you.